Good evening, everyone. Oh, that was nice. I'll do it again. Good evening, everyone. Oh, that's fabulous. Welcome. I am Elizabeth Herron, and I have the distinct honor of leading the advancement team at HudsonAlpha. We are delighted to have so many of you with us tonight. There are over 500 people who registered to come to this event with Dr. Neil Lamb. No real surprise, though, I'd say. For approximately the next hour and 15 minutes, you will hear amazing information and stories about how HudsonAlpha is doing work that impacts our community, our state, and really the entire world. But first, I want to say a special thank you to our sponsors who have made this event possible because it's free for the public, and our sponsorships help ensure that. So I want to thank our premier sponsors, the Ekta Foundation and the Mabubani family, and I want to thank you. Absolutely. And I also want to thank Paula Cushman, and I didn't see Paula out in the lobby. She may be here somewhere. There you are, Paula. Thank you as well for being a sponsor of this event. We appreciate it. Okay, now it's my pleasure to introduce Dr. Neil Lamb. So as many of you know, Dr. Lamb is the president of the HudsonAlpha Institute for Biotechnology, as well as an incredibly gifted science educator. Before becoming our president, Neil was the vice president for educational outreach and established an education program that has reached millions of students, teachers, and lifelong learners, and that's what we all are. I'm just wondering, I asked this a couple times we've done this, but who's attended a Neil Lamb event? Biotech 101, 201, anything else? Everybody look around. You see there are lots of hands up in the air. So that's wonderful. Well, I know you're going to enjoy what he has to share tonight, so without further ado, ladies and gentlemen, please give a warm welcome to Dr. Neil Lamb.
Thank you. Good evening, everybody. Woo, that is, wow, that's a powerful sound. It is so great to be with you tonight. I am so happy to be on stage. Thank you for spending some time with me. I know that you are busy, and there were probably lots of other things competing with your schedule tonight, but you are here. I also want to say thank you to the Mabubanis, and thank you, Paula, for making this possible for all the rest of us. We so appreciate and value your friendship. At the core of HudsonAlpha's mission is the pursuit of knowledge that transforms lives. Now, this spans all of our mission areas: research, economic development, and education. Tonight, I'm going to focus specifically on updates from our research. But I want to note that HudsonAlpha's value extends beyond research. It extends beyond the lab. Our economic development team fosters innovation, strengthens the local economy, and builds an ecosystem of practical output. Our commitment to education and outreach ensures that knowledge is shared broadly, cultivating the next generation of curious learners, and fostering a culture of innovation and curiosity and lifelong learning, like all of you are here doing. These teams are a critical part of HudsonAlpha. Now, I'd like you to think back on your day. I'd like you to think back on what your day was like. What all has happened since you first opened your eyes, since your feet first hit the ground and the sun came over the North Alabama foothills? What kind of a day have you had? Has it been a great day? Has it been a day that was challenging? Did you receive phenomenal news? Did you maybe receive not so good news? Did you get everything you wanted to get done on your to-do list today? Or was there something, many of you are just like me. Or were there things that you wanted to get to that you just weren't able to? Are there things that weighed you down today? Worries that you carried with you tonight? Now, I want to ask you to think about today for your family, for your friends, for your neighbors. What was their day like? Did they have challenges? Did they have phenomenal news? Did they have a celebration? Did they have something of a concern that they carry with them? We all have todays. And sometimes our todays are phenomenal, and sometimes our todays are not so phenomenal. I'd like to pull back even further, and I'd like to think about the 8 billion people that live on our planet and what their todays were like. What were their sorrows and joys, opportunities and challenges? Throughout tonight's presentation, I'm going to be giving you a handful of statistics about the challenges, the todays of our world. I'm not trying to overwhelm you with numbers, but I want you to have a sense of the common themes that all around the world, people like you and me face. The global nature of some of those challenges. And I want to do that because I want to show you how Hudson Alpha is working to solve those challenges today to make for a brighter tomorrow. So, with that in mind, let's get started. Around the world, on average, now again, this is average, I did not go count all this up. Around the world, on average, there were 385,000 babies born today. Now, for context, the city of Huntsville is about 522,000 people. So, about 2/3, 3/4 of Huntsville was born today and born every single day. Now, here's some interesting statistics that also go along with that. It's estimated that globally about 3 billion diapers were used today. It's also estimated that globally, about 5 billion cups of coffee or hot tea were consumed today, probably by those people who were taking care of these small individuals, these small children whose diapers they were changing at 3 o'clock in the morning. 385,000 babies. I want to note that about 3% of these, so about 11,000, were born with some sort of developmental or intellectual delay or rare genetic disease. And of that 3%, many of those were not identified at birth, maybe identified much later in life. Today, in America, 2,500 children received a diagnosis of developmental or intellectual delay or a rare disease. 2,500 today. Many of the families of these individuals waited an agonizingly long time to get that information, which fortunately is where Hudson Alpha comes in. I'd like to introduce you to two amazing young people. This is Jovi Cox on the left, and this is Cash Kennemer on the right. Both Jovi and Cash were seen at Hudson Alpha's Smith Family Clinic for Genomic Medicine, which is literally just right down this street right here. If you aren't familiar with the Smith Family Clinic for Genomic Medicine, the clinic uses genetic testing to identify rare and undiagnosed disease. And it's staffed by an amazing team of genetic counselors and a medical geneticist, Dr. Anna Hurst, who we share her time with UAB. The clinic is a lifeline for families that are seeking answers. After being seen at the clinic, both Jovi and Cash underwent whole genome sequencing, so their entire DNA was analyzed. And in both cases, genetic mutations were found. Jovi is actually the youngest reported person to be diagnosed with a disorder called Usher syndrome type 4. And Usher syndrome type 4 specifically is a condition that causes hearing and vision loss. And Jovi has moderate hearing loss, no vision loss at the moment, but it's likely as she gets older, she will have additional hearing loss and may experience vision problems. So early intervention is crucial for Jovi for optimizing care and her likelihood of the best possible outcomes. And her family had been looking for answers for a significant window of time before they came to the Smith Family Clinic. Cash was born with not one, but two genetic disorders. One linked to a gene called MAF and one linked to a gene called PTEN. You don't need to worry about what those specific things are, but Together, these lead to congenital cataracts, and Cash had cataract surgery when he was an infant. It also leads to developmental delays and an increased risk of developing cancerous tumors later in life. These disorders will be with Jovi and Cash for life, but genetic testing has empowered their families with a clearer idea of what the future holds, and both of them have now been able to access life-changing resources and develop long-term care plans because they got answers at the Smith Family Clinic for Genomic Medicine. Jovi received her diagnosis thanks to a partnership with the state's Children Rehabilitation Services. So this is a statewide effort working with children with special needs. The Smith Family Clinic is the clinic of choice for CRS, and we actually are getting ready to launch a telemedicine service so we can reach children in all parts of the state through CRS. Cash was brought to our attention because his family moved from California to Huntsville, and he was referred to the clinic for additional genetic testing. Many of the patients that come to the Smith Family Clinic qualify for financial assistance from a program called the Hero Fund. And I know that some of you in our audience today have provided support to the Hero Fund. That allows for these families to have this genetic testing without an undue financial burden placed on them. So on behalf of those families, thank you for those of you that have provided that support to the Hero Fund. It is a game changer for families not to have to worry about the financial costs of seeing a genetic counselor, a medical geneticist, and having genetic testing done. So let me transition, let me build on that. This is Dr. Greg Cooper, who is a member of the HudsonAlpha. He's a faculty investigator at the Institute. And Greg knows a thing or two about rare and undiagnosed diseases. Over the last decade, Greg and his research team have analyzed 2,000, don't take your time to count those, just trust me, have analyzed over 2,000 individuals with rare and undiagnosed genetic disorders. Sometimes that's been through the Smith Family Clinic, sometimes that's been in partnership with UAB or with neonatologists in Alabama or in other states. But Greg and his lab are dedicated to finding answers for families who lack information. His lab's goal is to discover genetic variants that lead to rare diseases, especially those that affect young children. And this is a crucial step, both short-term and long-term care for these kids. In nearly a third of these 2,000 patients, Greg and his team have identified the genetic change that causes the disease. That's an amazing set of people that now have answers where they did not have answers before. And maybe that answer actually changes patient care. More often, frustratingly than not, it doesn't. But it does provide an answer. And it provides a network for families to connect with other people who have similar genetic disorders. In another 20%, Greg's identified what's called a variant of uncertain significance. So this is a DNA change where we don't have enough evidence to be able to say, yep, this is causative, or nope, this is really not. The science hasn't caught up yet. And as the science progresses, Greg and his team continually go back and mine the data, and multiple times a year they're able to say, oh, yep, here's an answer that we found. We can actually now give this family an answer. We can move them from yellow to blue. But you see a huge number, half of these families, half of these families have no answer. And that's incredibly frustrating for the Cooper lab. So they are continually looking for new tools and technologies to help them provide more information to these families. Greg has recently collaborated with two other HudsonAlpha faculty investigators, Dr. Jane Grimwood and Jeremy Schmutz. They run the Genome Sequencing Center, and they have more than 2 decades of expertise in working with sequencing genomes of individuals and of multiple organisms. Jane runs the lab that actually does the sequencing work. Jeremy leads all the computational pieces. And in the last couple of years, an additional technology has come along called long-read sequencing. I talked a little bit about long-read sequencing if you were with us last August as one of the emerging trends that I thought would shape the field. HudsonAlpha bought 2 long-read sequencing machines last year using funding that came from the Innovation Fund. That's a special fund that we established to celebrate our 15th anniversary, and many of you in this room gave to the Innovation Fund. It's specifically designed to support emerging opportunities. So with those 2 new long-read sequencers, we did a pilot project looking at a small subset of these families that didn't have answers. And in a significant number of them, about 10%, we found results. So Greg and Jane and Jeremy just recently received news that a multi-year large-scale grant has been funded from the National Institutes of Health that will allow them to go back and sequence every one of those families that don't have answers. Now, we think that that's probably, on a conservative side, is going to give us maybe 10% more information, maybe even more. We aren't quite certain. So again, now we'll be dealing with 40% without answers, but every time the technology comes along, HudsonAlpha is looking for ways to apply that to glean more information for these families. Now I want to tell you about one additional, what comes next. And that's Dr. Andy Kadani. And I'm not sure if I saw Andy in the audience or not. I thought so. I think he's actually standing up at the back. Andy is the newest faculty member from HudsonAlpha. He joined us in July from St. Jude Children's Research Hospital. Would you help me welcome Andy? And Andy's goal is to understand disease mechanisms and develop therapeutic strategies that directly impact clinical care. Andy is passionate about families who have rare disease who have no answers for what they can do to try to improve life. And so Andy works with children, gets a genetic diagnosis, or maybe works with children that already have a genetic diagnosis, and then his goal is to understand the biology behind the pathway. So what is it about this mutation that changes the function of a gene, that changes the way it works? What else works alongside these proteins? What follows in the pathway? And then to think about, given what I know about the biology, can I begin to think about therapeutics? Maybe those are from existing medications that have already been approved. Maybe those are investigational medications. He's doing a lot of work right now helping generate enough preliminary data that families can actually enroll in and qualify for clinical trials in progress right now. And then in some cases, Andy actually looks to identify maybe there's something even more straightforward that we can do, like nutritional supplementation or fortification with some food that actually functions in the pathway that's disturbed that actually can make a difference. It's amazing and inspiring to listen to Andy. He has an unparalleled passion for children to make their life better, and he has a huge focus to do that specifically with Alabama kids and to make their world a better place. He is a perfect addition to HudsonAlpha and is the next stage in our ongoing commitment to working with families that have rare and undiagnosed disease. If you haven't met Andy, I hope you get the opportunity to do so. I'm not going to ask Andy to stay because he would be mobbed for an hour and a half afterwards, but if you get the chance to say hello to Andy, please do so. His work is amazing. All right, let me shift gears just a second. I have a riddle for you. Reach for the sky, yet anchored I stay. To bring forth life, I need water and clay. What am I? Now, I want you to think about that a second. Don't shout it out yet. Okay. What do you think this is? If you said plant or tree, you're absolutely right. So as we think about the importance of plants to our world, number one, plants make oxygen. You and I kind of like that. That's useful. Plants take sunlight and store the energy in sunlight in the form of sugar, and they provide the food that they either turn that into grains or fruits or nuts or seeds or things that are eaten by the things that we in turn eat. So really, plants sit at the kind of basic level of why we are able to have life on this planet. And plants are a really important part of the work that takes place at Hudson Alpha. Today, around the world, over 1 billion acres of land is cultivated. 1 billion acres. Now that may be for crops, that may be for fruit trees, that may be for berries, that may be for foraging crops for cattle. But 1 billion acres of land are dedicated to crop cultivation. I want to tell you 2 stories about Hudson Alpha's work in crops. The first is with peanuts. If you took all the peanuts that are harvested around the world in a given year and then averaged them out by 365 days, you would get 300 million pounds of peanuts a day. Yeah, that is a lot of Snickers bars. 300 million pounds of peanuts on average. That number is music to the ears of faculty investigator Josh Clevenger. He is such a fan of peanuts. And I asked him why he loves peanuts. And this is what he said. I want to read this to you in his words. Peanuts are a cheap, nutritious food that is accessible to even the most impoverished communities. And just 8 grams a day reduces the risk for heart disease, certain cancers, reduces bad cholesterol, and improves cognitive function. Side note, those of you that chose the peanut butter cookie tonight, bonus points for you. Peanuts use about 8 to 10 times less water than compared to other nuts, making them a more sustainable source of nutrition. And because they're actually a legume, they nourish the soil with nitrogen. Peanuts are the bedrock of one of the best products to treat malnutrition in the world because children like eating peanuts compared to soy-based alternatives. Peanuts are a major cash crop in many countries, including the United States. But in developing regions, peanut cultivation is a critical source of income for smallholder farmers and rural economies. I never knew peanuts were so important until I met Josh and until I listened to him talk about peanuts. And I am now a peanut fan because of all of these different components. And Josh is one of the leaders around the world in terms of thinking about peanuts and how to develop and breed better peanuts. Josh's lab harnesses genomics, so specific changes in those genetic recipes that lead to slightly different traits, maybe better use of water, maybe more resistant to disease, maybe a different profile in the amount of oils that are in the nuts that allows them to have a different taste or a longer shelf life. His work attempts to breed peanuts with increased heat tolerance, nutritional content, resistance to the effects of aflatoxin. I don't know if you know what aflatoxin is, but aflatoxin is a mold that grows in lots of different on lots of different foods. It's often found in the soil. It's not always as evident as what I'm showing you here, and there are people all around the world that unknowingly are exposed to aflatoxin in the foods they eat, especially peanuts. And it leads to long-term damage, including multiple forms of cancer and other disorders. It's estimated that today, 68 people around the world died because of long-term aflatoxin exposure. And many of those were children in third world countries. So you can see why Josh is focused on how do we breed peanuts that are resistant to aflatoxin. He also works on breeding peanuts that are resistant to peanut smut. Now, peanut smut is also a fungus, and that fungus gets into the shell. Remember the shell. Peanuts develop underground. The whole story of a peanut is fascinating. If you haven't heard Josh talk about it, I mean, I could wax poetic about peanut anyway. But because it develops under the ground, many of these fungal spores hang out in the soil, and they can stay in the soil for years and years and years. And they get into the peanut, and instead of forming the nut, the seed, it's just a whole ball of fungal spores. And you open the shell, and now we got it everywhere. So there are lots of countries that are battling the issues around aflatoxin and peanut smut. And Josh is on the forefront of breeding peanuts that are resistant and integrating that really into varieties around the world. Some of the work that Josh is doing is taking place in the wiregrass through our partnership, HudsonAlpha Wiregrass, with the city of Dothan. This is an incredible project. I hope you've heard a little bit about it. Josh is working directly with high school students. I think we've worked with 600 students so far over the last year and a half, and this week we started in 10 high schools across the region. Not new, but 10 high schools. These students are actually analyzing the DNA of a peanut that they are given that they grow, and then they take one of the leaves and they extract DNA from it in a protocol that would make a graduate student's head spin. These are high school ninth graders. We sequence the DNA and give them back some analysis to show them their peanut seedling, what are its superhero strengths and what are its Achilles heels. And based on their conversations with local farmers, the class makes decisions about which peanuts they think should be bred to create the best variety for what their area farmers need. They show their interest because we give them peanut butter bucks, fake money, and they buy. I would put my amount of money on this pairing, and then we do the breeding. One of the first trials, one of the first plants that came out of that, which a ninth grade student named Bruce, the student named the plant Bruce. The student's name is not Bruce, he named the plant Bruce. And Bruce is a phenomenal peanut, and Bruce is about to go into field trials, which is amazing. Developed by a high school student in partnership with Josh and with the education team at HudsonAlpha. But Josh's work extends beyond what I've just described. He actually is looking at multiple different plants and using these same genetic tools and analytical approaches to try to identify and improve different varieties. He's a prolific collaborator working with scientists and breeders in 19 countries across 4 continents. It is an incredible privilege to have him at HudsonAlpha. Another prolific collaborator is Dr. Alex Harkis. Alex is also a faculty investigator at HudsonAlpha. Alex works on plant reproduction and flowering. And let me preface what his research is by telling you that today, 12 billion pounds of fruits and vegetables were harvested around the world. So if you thought 300 million peanuts was a lot, 12 billion pounds of fruits and vegetables. That is a whole lot of kumquats. Alex has been studying the process of plant reproduction at the Institute since he was a freshman in college. So he was one of those that got turned on right out of the gate when he went to college and has never looked back. Last year, he received the National Science Foundation's most prestigious award for early career scientists. It's called their Career Award. And that grant, that Career Award, is actually generating the largest ever study of the genes that control plant reproduction. And if you wonder, well, okay, plant reproduction, so what? What does that mean? Reproduction is the foundation for all aspects of plant breeding and food production. No flower, no food. So understanding how plant reproduction and flower formation works is really important. About 10% of flowers in the world, about 10% of the species, actually come in male varieties and female varieties, not together in the same plant. And so you can take advantage of that understanding because you can use that information to identify genes. For some plants, the male plant may be what you want because it produces bigger fruit. Or the female plant may be the longer-lived producer. But you want to be able to know that early on, and if you can figure out what those genes are that control sex in plants, you actually can think about using that in plants that traditionally have both male and female on the same flower. You can think about how you breed that in to be able to create hardier, better, more food-producing varieties of plants. For many crops, Alex has quickly improved their entire breeding program by applying these tools. Beer hops, fiber hemp, apples, pear, asparagus, papaya, and more. He's also developing maturity biomarkers. So how can you tell when a piece of fruit is at its peak ripeness and should be picked. In some cases, one day too early or one day too late makes all the difference between the way the fruit tastes and how long it is shelf stable. The work that Alex does actually is critical to thinking about how we continue to feed a planet with a growing population when we have less and less farmable land to be able to use. And it's exciting to see the way that he is applying his career grant to push back the boundaries of understanding how plants reproduce. All right. 3 more stats for you. See if you can figure out how they're all connected. Today, 11,000 new automobiles rolled off the assembly line. In America, today in America, 1,500 new homes were completed under their construction. And today in America, 2.4 million pounds of packaging food waste were thrown away. Those are everything from the container, the clamshell that your strawberries came in, to the plastic film that sits over your meat, to takeout containers and plastic bottles. What do you think the connection is between all 3 of these? So let me give you a hint, because I've just heard some of you say that. And I will apologize in advance for what I am about to subject you to. Come and listen to my story about a man named Jed. Poor mountaineer, barely kept his family fed. Then one day he was shooting at some food, and up from the ground came oil, that is, black gold, Texas tea. Well, you're welcome, because you will not get that out of your mind for days. But all 3 of these have components that are produced by petrochemicals. So they're heavily dependent upon oil and gas. Whether those are the plastics in the cars, or the shingles and asphalts, shingles and cement and asphalt in the homes, or the plastic film or the plastic packaging. The challenge with those products is that oil and natural gas are limited resources. So the search is on to identify alternatives that are made from renewable, more environmentally friendly pathways. This is a story that I am so excited about, because I just find it amazing and a beautiful example of the kind of innovation that is taking place around our world and that Hudson Alpha sits at the center of. So we're going to start with grasses. This is miscanthus. This is a variety of miscanthus called Miscanthus giganteus. It grows 12 feet or more in a single growing season. Switchgrass is another grass that we're going to talk about. The beauty of these perennial grasses is that they can be started in marginal soil. And once you get them established the first season, they don't need a lot of water and they don't need a lot of fertilizer. And year after year, they produce this biomass, these shoots and leaves. And there's a growing field that is turning those shoots and leaves into products used by car makers, home builders, and food packagers. At Hudson Alpha, this work is led by Dr. Kankshita Swaminathan, Jeremy Schmutz, and Dr. Sam Jackson. Kankshita has extensive experience in working with these grasses and in studying how their genes impact the way they grow, the way they take up nitrogen, the way they respond to their environment. She is the grasses queen in many ways. I've already told you a little bit about Jeremy. Jeremy has 2 decades of computational experience in working with complex genomes, initially with human genomes, and now more recently with plant genomes. And Sam, Dr. Jackson, has both an academic background, a PhD working in grasses, and is the co-founder of a company that actually turns plant biomass into food packaging products. And so the goal, the project that they are working towards, would take this kind of biomass from perennial grasses that can be grown on marginal farmland. So number one, it provides another crop for farmers to grow on land that right now they can't do anything with. The beauty of these grasses is that they capture carbon, so they decarbonize an industry. They store that carbon in the soil, and they actually improve the soil's fertility over time. You can cut and bale these grasses, and then you take them to a processing plant that ideally you would build in the same community where you're growing these grasses, so you're not spending hours and hours transporting them by truck across the country. And you shred it into kind of what's called a feedstock, a starter material, kind of fluff. Now you can use this fluff as is for chicken bedding, but it can be used for so many more things based on what you then do next. Because this fluff is essentially carbons, hydrogens, oxygens, and nitrogens. And you can chemically adjust some of that, and you can use that to create fiber-based plastic alternatives. So for automobiles, you can create fiber-based dashboards and side panels, foam and fabric in your seats. For homeowners, for homes, you can use this as you think about insulation, as you think about shingles and asphalt and concrete. And for food packaging, you can turn this obviously into takeout containers, because that's what Sam's company does, but also for the packaging film, the flexible packaging, and for the medical packaging. I don't know about you, but my medicines come in so much plastic. So much plastic. And if that could be biodegradable, if that could be made from natural fibers, what a difference that would make. So this is creating what many of you, this is a phrase that many of you will be familiar with, a circular bioeconomy. The concept being that within a community, you are growing these grasses, you are harvesting them, taking them within the community to a place where you are processing them, manufacturing the materials that you need, using them, distributing them, and then based on the way you design the materials, based on the genetics of the grasses that you use, you may have a product that returns back that decomposes quickly, or you may have a product that decomposes more slowly based on what you need. You think about, we often hold a plastic cup or a styrofoam cup that is meant to hold liquid for maybe an hour or two, but it takes decades to break down. So if you could create something that would hold that liquid for as long as it was needed, but would more quickly break down, but then using other genetics of plants, you could have something that you need to last significantly longer, like the shingles on your roof. You don't want that to decompose in a matter of weeks. You want that to last for a matter of years. And all of that can be done by tweaking the chemistry and the genetics of the fibers of the plants that you're using. So here are some of our collaborators. And a lot of these names are things you're probably going to recognize, either involved in automobile or in construction or in transportation. FedEx, if you think about it, if we could figure out a way to make packaging that is just a gram or two less, when you have a plane or a truck full of packages, that less weight means less weight on the plane, less energy that's needed, but you're still getting the same amount of packages, the same number of packages. We are right now, HudsonAlpha, the hub for this group. So we have pulled all of these different players together, and we are competing for a National Science Foundation seed grant award of $160 million. Yeah, $160 million spread over 10 years. That is not the kind of change you're going to find in the cushions of your sofa. And the goal is that if we receive that, and this is going to be a multi-stage competition, so it'll be a while before we know, but if we receive that, we will be able to transform this part of industry in rural Tennessee and Alabama. It ties in with all of the automobile manufacturers in our two states, with the construction, the packaging, and again, HudsonAlpha and HudsonAlpha Innovation sit at the center of it. There's a workforce development component, there's an education component, there's an industry piece. It fits beautifully with the research, economic development, educational outreach mission of HudsonAlpha, and is right at the center of how we use today's discoveries to make tomorrow better. Does anybody in here have a birthday today? or yesterday or tomorrow. We'll extend that a little bit. All right, we've got a handful of folks. Okay, I'm not going to ask you your age, but if you happen to be 60, which you may not be, I think you're both in your 30s, but if you are 60, today you share a 60th birthday with about 65,000 people around the globe. So 65,000 people worldwide turned 60 today. Now, intriguingly, and it may be coincidental or not, today 65,000 pounds of Metamucil were also consumed. We're just going to assume that that's just by coincidence. But now let me share with you some numbers that are associated with becoming more mature, with turning older, that are not at all something that we would chuckle about. Today, 1,800 women received that call that said it's ovarian cancer. 1,800. Some of you in this room may have received that call in your life or have loved ones that have received that call. One of the challenges about ovarian cancer is that it is often diagnosed late and it is often very difficult to treat. It becomes resistant to treatment. This problem bothers faculty researcher Sarah Cooper. And Dr. Sarah Cooper does a significant amount of work looking at ovarian cancers, specifically looking at why certain ovarian cancers are not responsive to medications when others are, and what happens when those ovarian cancers become resistant to treatment. So she and her team have completed a screen looking for genes that are involved in this process of different response to therapy. And this summer, they started to dig into the function behind each of those genes. Some of those genes seem to control how medicines are let into the cell, or if they're allowed, or how they're kept out of the cell. Some of them seem to be involved in immune function. The exciting thing is that each gene represents a potential target for improving patient response, for chemotherapy, or maybe even an approach to thinking about a new drug. If you have been part of a Tie the Ribbons luncheon at Hudson Alpha, the support from that luncheon goes, among other things, to support the work of Dr. Sarah Cooper, both in breast and ovarian cancer. So if you have been to one of those phenomenal luncheons, thank you, because your support makes a difference as we try to find ways to change the pathways for those 1,800 women who got that phone call today. An even larger number, and this one really took my breath away. 31,000 people today were diagnosed with a neurodegenerative disease. 27,000 of those are Alzheimer's. Another 3,300 are Parkinson's, and the remainder are amyotrophic lateral sclerosis and Huntington's disease. 31,000 today. I don't have to tell you about the importance of finding ways to bring this number down and to offer more impactful treatment for those that have had diagnosis. And so I want to tell you two stories about the work of Hudson Alpha, specifically in this region. But in order to do that, I need to take us a little bit deeper into the science. So this is that moment in every one of our talks when I say buckle your seatbelt, because we're going to dive a little bit deeper. If this makes you go, oh, I feel like I'm back in high school biology, then you can close your eyes and go to your happy place or go get another cookie. I will not be offended. But I want to set up the science to help you understand the stories that I'm going to tell you about. You've probably heard me say countless times that DNA serves as a recipe, a set of instructions for how the cell makes something that it needs. Oftentimes, this is protein like insulin or hemoglobin or collagen. But the process of going from the genetic recipe to the actual product begins with first making a temporary copy of this genetic recipe, a short-term copy that goes out into the rest of the cell where it actually is then used as instructions to build the protein. That short-term copy is called RNA. We don't need to worry about that. But you often hear people talk about, or hear us talk about, turning genes on and off, or activating or silencing genes. That process of regulating gene activity is critically important because it is making that temporary copy that then allows us to make a protein. So if gene regulation is thrown off, if I have a gene that is silent when it should be active, I am not getting the protein I need. In the same way, if I have a gene that is active when it should be silent, I've now got something that's not supposed to be there. And then I can have times when my genes are producing at too high or too low of a rate of expression. So gene regulation is critical. It sits at the forefront of one of the biggest areas of scientific discovery. We now know the sequence of the genes. We now have the tools and technology to figure out what happens when there's a genetic change and to be able to detect it. But what's that mean functionally is something we're still trying to pursue. Understanding that is a huge opportunity for the field. So let me take us a little bit deeper. Okay, I'm going to show you a series of videos, and this looks like a telephone cord. Most of you here are old enough to know what a telephone cord is. If you're younger and don't remember telephone cords, teleties, which are the things that you can put around your hair, the all tiny spiral, that's the same sort of thing. But this red cable is DNA. And this piece that I've highlighted in green is a gene. So it is a set of instructions that tells the cell how to make something. Right, I guess I'll call it upstream, but at the top, you see this kind of fuzzy blob. That's a protein, but it's called a DNA binding protein. It binds DNA, as its name suggests. And it binds that DNA at a very specific sequence that I've shown here in yellow. So there's a very specific combination of A's, T's, C's, and G's that this binding protein specifically looks for and latches on. There are hundreds of DNA binding proteins in our cells. Each of them has a different little recognition sequence in your DNA that it looks for. All right, everybody with me so far? Ish? No, not yet. No. The key is that binding protein is really important to activate, to turn on the gene to make that copy that we need to make protein. So let's turn the video on. Okay, so you're going to see this protein. You're going to now get a different view where, whoop, here comes another binding protein, actually a whole complex of them. And so it's linked in place, and then a whole bunch of other ones come to the table just as well. It's like a pileup on I-565. And so they're all together now, each of them bound to a different piece of the DNA. And this is a part of the complex that's actually needed to make this copy. And then at the very end, you saw this DNA kind of loop around with this white blob. That's another DNA binding protein. These regions can be thousands of A's, T's, C's, and G's away from the beginning of your gene. It's fascinating. Your DNA does all these contusions and contortions and yoga to bind, to get all the right pieces in the right places. So what's going to happen when this white binding protein connects with the larger complex is it sets free the machinery that is going to zip along the DNA and make a copy. Now, this is going to move really fast. It's going to move at the speed, the number of letters copying as it is happening inside your cells every minute of the day. So here we go. And we're off. Okay, so we are zipping along, and that yellow piece that's trailing is the copy that's being made. So super fast. Here we are. You can see that we've unzipped the DNA. This machinery is zipping through. These little yellow pieces are being added in, building out that RNA copy that is streaming out the back. Woohoo! Here we go. Boom. Okay. That is happening inside every one of your cells hundreds of times every second of every day of your life. That is fascinating. Absolutely fascinating. And that's the process that makes you and I sit here enjoying our cookies and engaging with each other. But it all starts with bringing all the right pieces together to activate that gene. So gene regulation, to understand how genes are activated, we need to know the specific control regions, those sequences in and around each gene, and the proteins that actually bind them. I'm going to call them the control switches. So I've got to know the control regions, that's the DNA sequence, and the control switches, those are the proteins. Everybody with me? Okay. Now I told you, knowing this is the secret to understanding gene regulation. This is about as deep as we're going to go. I'm about to pull us back up. So everybody, if you stepped away mentally, come back and engage with me again. I told you that understanding the process of gene regulation is one of the next frontiers for our field. Fortunately, one of the leaders in exploring gene regulation is on the HudsonAlpha team. In fact, he is sitting in the room in the back corner. Our Chief Science Officer and President Emeritus, Dr. Rick Myers. So how do we apply this to neurodegenerative disease? How do we think about what this means as we look at what is happening inside the cells of the brain? What is happening with that process of genes being turned on and off and the different signals going back and forth? That's some of the work that Dr. Myers has put together. In fact, Rick and his lab have had an incredibly productive year or so in research. In the last 14 months, they've published 6 papers specifically done primarily from work in their lab, and another 10 in which they were significant contributors. 5 of those were in collaboration with other HudsonAlpha faculty. Rick is also a prolific collaborator. The number of papers you publish is a key measure of the strength of your science, but so is the amount of grant funding that you receive. So these publications have actually led to 3 new funded grants and several additional applications that are pending. Specifically, what Rick and his team are doing is building a database of these control regions and switches in the human brain. So they are working with multiple regions of the brain from post-mortem brains from donors, and they are looking in each of those regions. They separate it out into the different types of neurons and supporting cells that are in the brain region, and in each of those, they are looking to see where are the control regions and where are the protein switches binding to those control regions. So they are building a database of the brain's regulatory gene regulation process. The Myers Lab and their collaborators have actually built the largest repository of this information to date in the world. So they hold the largest amount of information related to how genes in our brain are regulated. And this is incredibly valuable as we think about not only typical brain function, but as we think about neurodegenerative disease and neuropsychiatric illnesses, because we can begin to look for ways that those regulation patterns change. Now, this research, I told you Rick has gotten several new grants. This research is also supported by funding from Memory and Mobility. And so those of you that have donated to the Memory and Mobility Fund, you've actually helped fund the pilot work that has led to a lot of this. So thank you for that. That is so incredibly important. A key area of the work in Rick's lab is exploring the relationship between neurological disease and DNA variation in the control regions and the genes that encode the switches. Now, you've heard me talk about DNA variation. It's simply changes in the recipe. And Rick's lab is looking at specific changes in the recipe in these regions that may or may not influence how genes are turned on and off, and the amount of on and off that they experience. If you think about it, it's not unimaginable that individuals with changes in some of these key regions might have different levels of gene activity in the brain and then different responses, different susceptibilities to cell breakdown or cell damage or environmental responses. So Rick's identified thousands of DNA differences that impact how this process works. And when I asked him to tell me why this work is important, here's what he said. My lab has already begun to apply this knowledge to test for responses to existing drugs and possibly new sets of compounds that are not yet drugs to speed up the development of treatments. We are excited by these approaches because we believe they will have a large impact. Because we have many years of developing and applying these techniques, we are in a unique position to make many of those impacts. So a significant amount of time invested in building the database, and now we begin to look at the functional significance and the functional meaning of that as it relates to neurodegenerative disease. Rick's work looks at lots of different genes. Another one of our faculty investigators, Dr. Nick Cochran's work, focuses on a smaller subset of genes. Specifically, Nick is working with the gene that produces the protein called tau. And this is an artistic image of a cross-section of a region of the brain from an individual with dementia. And the purple cells are the living cells, and that big clump of brown is a tangle of tau protein that is just gunking up the entire space around the cells. These tau tangles are a hallmark of many different kinds of dementia. And Nick is trying to understand, again, what are the control regions and the switches that drive the production of tau. So you can see the gene for tau in blue, and then you can see the surrounding DNA. And he's looking specifically, what are the DNA stretches in yellow, and what are the binding proteins? Nick's a relatively young faculty investigator, and his work, much of his pilot work, was also funded by Memory and Mobility. You may have seen in the news today, Hudson Alpha put out a press release about a new multi-year $3.5 million NIH project around this exact work that is Nick and Rick. Your dollars, your Memory and Mobility dollars, provided the pilot data that allowed them to write the successful grant to now study this work in tau in Alzheimer's. This is Nick's first grant, and it's a big deal when you get your first grant from the National Institutes of Health. And speaking to the incredible value of Nick, the intelligence of Nick, the way he thinks about projects, Nick is doing that. Nick has gotten his first grant at a significantly younger age than the average age of the first grant. I look at Nick and he feels like to me he's like just barely out of high school. He's not. He's older than that, but he is an amazing researcher. And if we can understand the regulatory regions that control how much tau is produced in our brains, then we can open the door to thinking about ways that we can reduce it or that we can control or regulate it. Nick will actually pursue similar kinds of projects for other proteins that junk up the cells and the area around our cells for neurodegenerative disease. Amyloid beta is a protein that you may have heard a lot about. It's found in the neurons, in and around the neurons in Alzheimer's patients. Many of the FDA-approved treatments that are brand new are trying to get rid of levels of A beta. And Nick wants to understand what it is that adjusts or changes the regulation of that. And then also for individuals with Parkinson's, there's a protein called alpha-synuclein that gums up the cells and the regions of the brain, and trying to understand how that's regulated. Speaking of Parkinson's, some of you are aware that we have a clinical biomarker trial underway at Hudson Alpha, enrollment through the Smith Family Clinic. We are looking for biomarkers, so proteins or other small molecules that are found in blood that we can use as an early stage diagnostic for Parkinson's or as a way to follow the progression of Parkinson's. Many of you in this room may have enrolled either as a case, an individual with Parkinson's, or as an age-matched control. And we are on track to meeting our enrollment numbers, so thank you for that. The last story that I want to tell you also deals with identifying markers to track disease, but on a much larger level. And to do that, we're going to have to peer deep into our crystal ball, because I am now taking us to the edge of the field of science. So, it deals with this really important question. How do we maintain health and prevent or slow disease? So how can we keep ourselves healthier for longer windows of time? So if you imagine in our lives, there's a three-stage phase. Health, then there's a stage where we begin not to be so healthy, and then we move into disease. And so those three stages, the goal, if we could figure out how to delay the onset of disease, then we would end up with something that looks more like this, where we have a longer window in the blue, a longer window of health. That's called our health span. We're not necessarily increasing our lifespan, but we are increasing the amount of our lifespan that we are healthy. And then a smaller window for transition and disease. And so I want to introduce you to something that becomes really important as we think about that. And it's a word I hope you'll be hearing me use a lot in the future. It's called the phenome. This describes the physical and biochemical characteristics of an organism. It comes from the word phenotype, which is a scientific word that we like to throw around that means anything that you can measure, anything that you can observe about an organism. In this case, about you and me. And I've got some images of some of the things that we can measure or that we can track. Our diet, the amount and type of exercise that we get, if we take nutritional supplements, how much sleep and the quality of the sleep that we get, measures of brain health like our ability to do crosswords and Sudokus, molecules that are present in our blood, saliva, or urine, again, biomarkers, our past health history. Phenomes are the interaction between our genes and our environment, and they are dynamic, constantly changing. So if you measure them today and you measure them a week from now or 10 minutes from now or 6 months from now, they may be different. And they may tell us something about the state of what's going on inside our body and about health and disease. So imagine if you could track dozens or even hundreds of these markers of your phenome. I'm showing some of them up here. There are so many more. What if we could follow them over time? So how do they change 6 months from now, a year from now, 5 years from now? Now imagine that we can do that for 1,000 people or more. Now we're beginning to get enough data that you can look for common patterns as people progress into certain diseases or as they move through different stages of health, disorder, and healing and return to health. That's the ultimate goal for what we want for these phenome studies, to find the biomarkers, to find the phenome markers that tell us when we have moved out of health before we ever develop symptoms of the disease, so that we can then identify and design a therapy that puts us back on the path to health. So if you are thinking that this is a massive data challenge, you are completely correct. Every one of those pieces of data, every time that it's tracked, for every person that it's tracked, goes into a massive database. Millions and millions of data points. And then you need to try to figure out, how do they all connect to each other? What's the pattern? What pieces are linked to each other? Artificial intelligence and computational modeling are going to be the things that help us figure this out. Looking through all this data, trying to find patterns, and then replicating those with new individuals and looking for those pieces over and over until we can say, okay, for a disease, for example, for dementia, for a certain form of dementia, here are all the factors that are associated with that we've tracked through people's phenomes. That could be specific protein markers that are in your blood. That could be your score on specific neurological tests. That might be the level of exercise or activity that you get. That might be something like blood pressure and heart rate. But here are all the pieces that are somehow linked. And then, because we would have done this for thousands of individuals over time, you could begin to say, this is often the first place that we see change. And then that progresses, and we see additional markers that say, hey, we are starting down the path out of health. Maybe this many markers is when you get the first physical symptoms, maybe even so mild that you don't even notice them, but the first signs, the first observable signs, even though we've had all of these phenome markers telling us things are off track. And then everything is thrown off by the time we're in full-blown disease. A phenome study a well-done phenome study, we believe can get us this kind of information. And then what do we do with it? Well, then you can work backwards and you can say, when I'm at this point, knowing the biology, knowing the function, what's a specific intervention that I can design? Maybe that's a nutritional supplement. Maybe that's a small drug molecule. Maybe that's changing the amount of exercise I get or ensuring that I am getting better sleep. I don't know what that would be. But you identify, develop, and apply an intervention that takes you back to a healthy pathway. That is what I hope we step into. That is what I think is a future project for science, and I think a future project that has a home at HudsonAlpha. There are a lot of challenges, as you might expect, to doing this. How do you recruit 1,000 people and ask them to track all these different pieces and maybe wear some sort of technology and maybe ask if they would give you a sample of their poop every 6 months so that you can look at the bacteria in their gut? And will they tell you the truth about what they ate today? Will they ignore the fact that that 4 bags of M&Ms, peanut M&Ms, just doesn't make its way to the dietary list? How do you recruit from a diverse population so everybody isn't exactly the same? Because we've got to have as many different people from as many diverse backgrounds as possible. How do you track that? What kind of system do you set up? And how do you pull that information out and then test it? How do you pay for that? That is a massive, massive project. But I think it's an incredible opportunity to partner with our community and incredibly bright minds and try to solve that problem together, pushing back the boundaries of our knowledge. So we've just covered a whole lot of scientific ground, a lot of content tonight. So let me summarize it like this. HudsonAlpha is not just a center of scientific research, but it is a lighthouse. It is a guiding beacon pointing us towards a better tomorrow. Every day, our dedicated scientists unravel the threads of DNA and push us towards further advancements in human health and agriculture. Our work is driven by this belief that every scientific breakthrough has the potential to touch lives beyond the lab. That is what we are here to do, to translate scientific discoveries into real-world applications that make the world a better place. To use the power of science to address some of the most pressing challenges that you and I and all of our global neighbors face every day. It offers a tangible promise of a future where every child has a chance at a healthier life, where farmers can grow crops that flourish despite uncertain environmental conditions and ravaging pests, and where individuals no longer face the fear of untreatable disease. And these advances are available to everyone. So as I close out my presentation, and as the sun sets on our today, I want to leave you with a final thought. Today's efforts within HudsonAlpha's research labs, spaces in our associate companies on campus, and in our educational spaces make for a brighter, healthier, more sustainable tomorrow. The power of a better tomorrow sits in our hands today at HudsonAlpha. And I'm incredibly proud to be a tiny part of that. And those of you that are in this audience that are part of the HudsonAlpha ecosystem, from a research lab, from an associate company, from an education space, from the supporting team that makes everything work, thank you. Because your work is what allows us to imagine and put into action brighter tomorrows. And for those of you in this room that have supported HudsonAlpha in the past, through the Innovation Fund, Memory and Mobility, through the Hero Fund, or through any other gift, you empower the ability for us to bring tomorrow into today. I and my entire team and the Institute are incredibly grateful for that. And with that, I will wrap us up and I'll take questions. So let me take just a couple minutes to see if we've got any questions, and then I'll bring Elizabeth up. Any questions? Oh, how did I create the movie of replication? I should have said that was created by a great organization called the Howard Hughes Medical Institute. They have a number of beautiful animations that are used in classrooms all around the world, and that's one of them. It's available on YouTube, and they give you permission to take it and pause it and add things to it and then run it. So HHMI is the place you want to look for that. Is there a way to prioritize the effort I described at the end around phenomes towards extending health span? So part of that means we've got to understand, what does healthy aging look like? And because there's no way that a 60-year-old and a 40-year-old and a 20-year-old are going to have the exact set of phenome measures. So we've got to understand what healthy aging looks like and be able to see, what do we begin to identify when we move into disease. I think, Ashok, that part of a phenome trial around disorders of aging would begin to give us some of those healthy measures as well. It's a great question. Yes, sir. Yes. So that article that came out a couple days ago talks about aging, that the rate of aging is not linear, that we age at at one rate, and then around the mid-40s, there's a rapid spurt in aging, and then it kind of levels out again until 60, and there's another rapid spurt. And then this paper didn't show it, but earlier papers actually found that there's another spurt in your 80s. Yeah, so that process, and I think, again, a phenome study would get us there. Okay, Elizabeth, come on up. Thank you again, everybody. It is a joy to be with you.
Okay. Am I on, Steve? There I go. Okay. So I think you just answered my question. Did you enjoy tonight's presentation? Would you like to see Dr. Lamb do this again next year? So for those of you that were here last year, he told me at that presentation he would do it again this year. So I think we just submitted, maybe we've got you on the calendar for 2025 for our next event series. Thank you. You know, we heard some really incredible stories tonight about how Hudson Alpha is impacting our world, our state, and certainly our community. And Neil mentioned several ways that philanthropy has played a role in making those impacts happen. He mentioned the Memory Mobility Program that's enabled new research studies in many types of neurodegenerative diseases. Talked about the Parkinson's patient enrollment study, which we're so excited about at the Smith Family Clinic. That was all made possible through philanthropy. Donations to the Smith Family Clinic help make it possible for us to diagnose those precious children that you saw on the screen tonight that have rare disease. He talked about research on peanuts, a peanut really, but so much of the world depends on peanuts as a critical source of protein. And so research being done on peanuts and smut disease, as well as aflatoxin. And then he pointed out the miscanthus and switchgrass plants that they are studying for the exciting Greening the Southeast program. All of those plants are grown in our greenhouse. And so many donations have supported our greenhouse. If you have made a donation, a contribution to support any of this work, please know sincerely how much we thank you and we appreciate you. You are part of our story, and we could not do what we do without your support. We are truly grateful. I hope you feel that, both from Neil and from me and really everyone at Hudson Alpha. We hope you feel inspired by what you heard tonight and will continue to share our vision of making better tomorrows. So, Neil said he was going to stick around after the program. He's right up here. If you have questions, if you want to talk to him, he'd stay as long as he needed to stay. My wonderful team, the advancement team, who are in the lobby, y'all wave back there so people can see you. They're back there. If you're interested in making a donation, we would certainly love to accept it. If you've never had a tour, it's been a long time since you've toured Hudson Alpha, please talk to a member of my team, and we can schedule you to come back for a tour. And then lastly, I want to remind you about two last events in our 2024 event series. One on Tuesday, September the 10th. It will feature Kelly East, who's our Vice President for Educational Outreach, and she will be talking about understanding your family health history. That will be here at the Jackson Center. And then our last event of the year will feature our special guest, Dr. Harold Varmus, who is a close friend of our own Dr. Rick Myers. It's coming at his invitation. Dr. Varmus, you may recall, is a former director of the National Institutes of Health, as well as the National Cancer Institute, and is a Nobel laureate for his discovery in oncogenes. So we are so excited to have him here, and that will be on November the 19th. Go ahead and put those dates on your calendar. You'll get emails and reminders about those in the future. So with that said, once again, thank you for joining us tonight. It's great to see you all. Have a wonderful evening.